Cerrahpasa Neuroscience Talks. Today, we have the privilege of hosting a truly distinguished neuroscientist whose work has significantly shaped our understanding of brain aging and neuroplasticity, Professor Dr. Mark Mattson. Today's seminar focuses on a timely and highly impactful topic, intermittent fasting and exercise for brain and body health. This subject beautifully bridges fundamental neuroscience with practical lifestyle interventions, highlighting how metabolic challenges such as fasting and physical activity can enhance brain function, resilience, and longevity. Before we begin, let me briefly introduce our esteemed speaker. Professor Mattson is an American neuroscientist and adjunct professor of neuroscience at the Johns Hopkins University School of Medicine. He previously served as chief of the Laboratory of Neuroscience at the National Institute of Aging, part of the National Institutes of Health from 2000 to 2019. He is internationally recognized for his pioneering research on the cellular and molecular mechanisms of brain aging and neurodegenerative diseases, particularly Alzheimer's and Parkinson's disease. Over the past decades, his research has explored how lifestyle factors such as intermittent fasting and exercise influence brain function and resilience. His work has helped illuminate how the brain adapts to metabolic challenges and how these adaptations may promote long-term health. With more than 230,000 citations, Professor Mason is among the most highly cited neuroscientists in the world. His work continues to bridge basic science with translational approaches aimed at promoting healthy brain aging. It is an immense honor to host him today, and we eagerly look forward to the insights he will share with us. Following the presentation, we will have a Q&A session. And now, without further delay, Professor Matson, welcome again. The floor is yours.
Thank you for the kind introduction, Talha. Okay, so this should be it. So, as you said in the introduction, I'm going to talk about research both in animal models in the laboratory and in humans in clinical trials, mainly looking at the effects of intermittent fasting and exercise. On health in general. I'm going to focus on the brain because I'm a neuroscientist and that's what my lab is mainly focused on. But of course, there's connections always between the body and brain. And in fact, some of the benefits, for example, of exercise and intermittent fasting on the brain are mediated by factors released into the blood from peripheral tissues. All right. So this first introductory slide just essentially, let me see, get this out of the way. What this does is an evolutionary perspective, just pointing out that during evolution, animals in the wild, including our human ancestors, had to deal with a lot of different environmental challenges or stresses, one of which was food scarcity. In fact, there's quite a bit of evidence that competition for limited amounts of food was a major driver for evolution of many different species. And in the upper left is shown some wolves surrounding a buffalo. And presumably somewhere at some point in evolution, uh, wolves that cooperated with other wolves to track, surround, and then eventually kill large prey animals had a survival advantage. So the social brain, in the sense of working together to accomplish a specific goal, was driven by food scarcity. And then in the lower left is hunter-gatherers in Africa. These are obviously modern-day hunter-gatherers, but this is the environment and lifestyle, if you will, that humans existed in. And from the standpoint of the brain and evolution, as far as we know, all the earliest tools invented by humans were tools that their function was to either acquire or process food. So things like flake stones, spears, bow and arrow, and so on. Okay, so fast forward to 2026, and in the United States and elsewhere, Uh, Overweight and obesity and associated diseases, diabetes, cardiovascular disease, are common, too common, not only in humans, uh, as a result of fast food and the companies that essentially manufacture and market foods that are addictive. And also pets, you contrast a domesticated dog to wolves, and not always, but in some cases, people overfeed their pets and they don't get much exercise, and so they're not good metabolic and brain health. In fact, if you take a dog, domesticated dog that's the same body size as a wolf in the wild, it's been shown that the brain of modern day dogs is smaller than the wolf. So there's actually been a devolution, I guess you might word it that way, devolution of these domesticated animal brains. And then from a historical perspective, and which is some cases relevant to Ramadan, as you know, most or all major religions fasting of one type or another is an important component of it to various extents depending on the religion and the individual, I guess. But going back in history, philosophers, for example, recognize the importance of exercise and not overeating for the brain. So here's just a few quotes here. Okay, so now I'll get to the science. I'm going to start with intermittent fasting. Intermittent fasting is not a diet, it's eating pattern. So diet have to do with what you eat and how much you eat, uh, not really so much when you eat. So this from a recent review article I wrote for Nature Metabolism. This is just kind of some of the definitions of the most commonly employed or deployed intermittent fasting eating patterns in animal studies in humans: time-restricted eating, compressing the time window one eats each day to, say, 6 to 8 hours, so that you're fasting up to 18 hours every day. Um, this caloric restriction with time-restricted eating, it turns out that, and I'll mention this in the next few slides, that there are hundreds and probably thousands, I guess, of published studies of caloric restriction in animal models, rats and mice. And it turns out that essentially all of the calorie restriction studies, if you go on PubMed and look at rats and mice, those were also daily time restricted eating studies, because the way the experiments were done is that the calorie restricted animals are given their total daily allotment of food all at once. And when they're getting far fewer calories than they would normally eat by themselves, they eat it all quickly. And so they're actually fasting for up to 20 hours every day. This becomes important because we're finding many laboratories, including clinical trials, are finding that at least some of the beneficial effects of intermittent fasting eating patterns can be dissociated from any overall reduction in calorie intake. So there's beneficial effects on health that go beyond what can be accounted for by any decrease in, for example, weekly calorie intake. Alternate day fasting or food deprivation. This is one uh, intermittent fasting eating pattern we used a lot in our animal studies. Alternate day modified fast. So these modified fast is essentially during the fasting period, you're eating only about, say, 500 calories. So for example, eating 500 calories on 2 consecutive days a week, and then eating as you normally would the other days. And then the control eating patterns are ad libitum feeding or what's called continuous calorie restriction, where, for example, in human studies, eat breakfast, lunch, dinner spaced out, but each meal has fewer calories than normal. So there's just some examples. If you take blood samples and measure glucose and ketone levels, this is a kind of the typical eating pattern. Breakfast, lunch, dinner, an evening snack. Every time you eat, blood glucose levels go up and then come down uh, as the glucose is taken up by the liver and other cells. There's no elevation in ketones, and in the next slide, I'm going to show you this kind of metabolic switch from glucose to ketones. With alternate day fasting, in this case, the individual is fasting completely for one day and then eating breakfast, lunch, and dinner the next day. And so glucose stays low during the fasting period, and ketones will go up typically about 10 to 12 hours after the onset of complete fasting. And this is time restricted eating on the bottom, compressing the time window to 6 hours. In this case, the individual does not eat breakfast. So ketones then go up in the morning hours, and then when they eat, ketones go down. Okay, so this is what's happening with fasting and also with extended exercise. Like if you go out and run for an hour, the same thing may happen. That is, the glycogen, the glucose stores in the liver are depleted. And then that triggers breakdown of triglycerides and diglycerides in the fat cells and the release of free fatty acids into the blood, which are then taken up by hepatocytes and converted to the ketones beta hydroxybutyrate, BHB, and acetoacetate. These ketones then are transported. There's specific transport proteins in the membrane of cells. For example, neurons have a ketone transporter called MCT2, monocarboxylic acid transporter 2. And then the ketones, acetoacetate or BHB, can be used to generate ATP. So they're an alternate energy source for cells. I'm showing neuron here. Also, this ketone, BHB, interestingly, acts as a hormone. It has signaling functions. It will activate signaling pathways in cells. For example, we found that it will induce transcription factors in neurons in the brain that stimulate the production of a neurotrophic factor called BDNF, brain-derived neurotrophic factor. And I'll mention this later. This is a very important protein in the brain. Its levels increase in response to physical exercise, to fasting, and to what you and I are doing now, activity in neural networks. So right now, we're having a lot of activity in our hippocampal neurons, the neurons involved in learning and remembering things. And BDNF is critical for learning and memory. It promotes the growth and maintenance of synapses between neurons. It also, in the hippocampus, stimulates neurogenesis, the production of new neurons from stem cells. Okay. Right, so this is just lifespan data from early caloric restriction studies done by Rick Weindruck, where they progressively reduced calorie intake in rats and found that this is the body weight, and found that it extended lifespan. So if you reduce by whatever 20%, you get this much increase in average and maximum lifespan. If you increased by 40%, 30 to 40%, you get this. So there's a big increase in lifespan when caloric restriction slash time restricted eating is initiated in young rats or mice. And these are the only interventions that consistently and robustly increase lifespan in laboratory animals. This slide just simply shows that with this typical calorie restriction studies, the animals eat all their food within a short 4 to 6 hour time window when you restrict. And this is shown using a different, more cutting edge recent approach. And this is a ketone BHB. It goes up with fasting and glucose is down. This is a study by my colleague Don Ingram at the National Institute on Aging. They showed that alternate day fasting, they called it every other day fasting, extends lifespan in rats by a lot, 50% in this study. And as far as body weights, if you compare the animals on alternate day fasting, this is done when they're young, they initially gain weight, mostly muscle mass, and then when they get to adulthood, they kind of stop gaining weight compared to the ad libitum feeding animals. So this weight difference is mostly fat between these two groups. I'm not going to go over any of the details of this of these images in this slide. These are just examples of data from studies where postdocs in my lab showed that if they maintain rats or mice on every other day fasting in animal models of epilepsy, in the upper left, stroke, upper right, Parkinson's disease, lower left, and Huntington's disease, lower right, neurons, vulnerable neurons are more resistant to dysfunction and degeneration, and there's a better clinical outcome. For example, in the epilepsy model, which damages these hippocampal neurons, these animals will get cognitive impairment normally, but if they're on the alternate day food restriction, there's protection of these neurons and there's preservation of performance in learning and memory tests. In the stroke model, there's less brain damage and improved functional outcome, etc. And then at that time, there weren't really any good models of Alzheimer's disease, but in the early 1990s, with the cloning of the gene for the amyloid precursor protein, which is the source of the amyloid beta peptide that accumulates in the brain in Alzheimer's, transgenic mouse models were made that express mutant forms of amyloid precursor protein or other proteins that cause early onset Alzheimer's disease in humans. Frank Lafleur, a colleague of mine, and with our help, made what's called a triple transgenic mouse model of Alzheimer's disease, 3x TGAD mice. And he used a really cool method to make these mice. We sent him our presenilin 1 mutant knock-in mice. Those are mice that have a presenilin mutation that causes Alzheimer's disease in humans. But those animals didn't get amyloid plaques because they didn't have the human amyloid precursor protein. So what Frank's lab did is they bred two mice and took essentially the one cell stage embryo, the fertilized egg, out of the female and injected it with two different expression plasmids, one with mutant tau and one with mutant APP. So then they implanted this fertilized egg with these mutations, implanted the embryo in the uterus of a female, and it gave birth. And so these mice had three gene mutations in them. So we didn't have to do any crossbreeding, which makes a lot of problems with the genetic. Uh, so anyway, these mice, as they age, they get amyloid plaques in their brain, shown here in green, and tau, hyperphosphorylated tau tangles in the hippocampus. And they have learning and memory deficits. We took these mice and beginning at 5 months of age, we randomly assigned them to ad libitum feeding, alternate day fasting, uh, daily calorie restriction / time restricted eating. And then we had non-transgenic controls, and we let them go for a year. And then we tested their learning and memory in the water maze. And what you can see is both the alternate day fasting and the daily calorie restriction ameliorated the deficit in learning and memory. Okay, so now in the title of my talk, I mentioned the body. So I'm going to briefly talk about effects of intermittent fasting on the body. One interesting effect we found in rats was that uh, after the initiation of intermittent fasting, alternate day fasting, over a period of several weeks, the resting heart rate and blood pressure of the rats decreases. And then through other studies using manipulations that are in these references here, we showed that what's happening is the intermittent fasting is enhancing parasympathetic tone of the heart cardiac myocytes. Uh, uh, so if you remember, the parasympathetic nervous system activation slows down heart rate. The sympathetic nervous system increases heart rate. So we're finding the intermittent fasting uh, enhances parasympathetic tone, actually by upregulating BDNF in the brainstem cardiovagal neurons. So that was interesting. What's also interesting in this is same general effect of endurance training, athletic training. So endurance athletes will generally have low resting heart rate, low blood pressure, and high heart rate variability because they have increased parasympathetic tone. In fact, we showed that this is just showing some of the raw data where we find intermittent fasting or daily calorie restriction reduce resting heart rate and blood pressure. And then if we switch them back to ad libitum feeding within a couple weeks, the heart rate and blood pressure goes back. So this is like not sustained after you stop uh, intermittent fasting. And that's kind of similar again to exercise. If you've been exercising regularly and you've got your blood pressure and heart rate down within a few weeks, certainly by a month, if you completely stop exercising, those go back up. We also showed that intermittent fasting protects the heart against a myocardial infarction. That's in this paper. So similar to the stroke in the brain, it's kind of the same mechanisms. We had evidence that the intermittent fasting upregulates cellular stress resistance proteins, including heat shock proteins, these protein chaperones, neurotrophic factors in the brain, antioxidant enzymes. Uh, right. So then I collaborated with several clinicians who saw our work in animals and were interested in intermittent fasting. First, we did a small study in asthma patients. We and others had shown that the intermittent fasting will suppress inflammation, for example, in the stroke model, but also in the heart. And so this clinician at Louisiana State University, he contacted me and said, can we collaborate on a study in asthma patients? So we did a study where it was a very small study, only 11 overweight asthma patients with moderate to severe asthma. And we took a bunch of data at baseline, their asthma symptoms, their airflow in the lungs, and blood samples. And then we put them on every other day modified fasting, where every other day they ate only 500 calories. And we maintained them for 2 months. And we found that first, they lost quite a bit of weight on this regimen, which makes sense if they're not on the feeding days, not overeating much. Um, their mood improved. They're losing weight. Maybe that has something to do with it. Maybe BDNF levels are going up in their brain, and their airflow increased, uh, and their asthma symptoms improved. And in the blood, we found uh, over a period of 2 weeks to a month, reductions in markers of oxidative stress and inflammation. Then we did a study in collaboration with Michelle Harvey in England. She works with women at risk for breast cancer because of their metabolic status, their overweight, and also family history. Many of them have mother or sister or someone relative that had breast cancer. And so in this study, we randomly assigned 100 of these women to either 5-2 intermittent fasting or continuous intermittent fasting, defined as I showed in that early slide. And we maintained them on this for 6 months. We found that both groups, with both interventions, and I should say both groups, their weekly calorie intake was the same. And we designed it specifically this way. So the group on continuous energy restriction, they ate breakfast, lunch, and dinner, but each meal had 25% fewer calories than they would normally eat. And then the women on 5-2 intermittent fasting, 2 days a week, they ate only 500 calories. But if you do the numbers, their weekly calorie intake for both groups is the same. And indeed, they both lost the same amount of weight. But the women on the 5-2 intermittent fasting had a greater improvement in insulin sensitivity, so the greater improvement in regulation of glucose. So their system is better able to remove glucose from the blood and get it into cells after eating a meal. Okay, that specific study led to a documentary by the BBC, which aired in 2013. And that in turn led to intermittent fasting becoming a thing on the internet. Before then, if you Googled intermittent fasting, mainly you'd get some research articles coming up. Now, if you Google intermittent fasting, it's hard to find the research. You get all these random things coming up. But there's been a lot of work in the last 15 years in particular, and it's accelerated during that time, on intermittent fasting in animals and human clinical trials. When I retired from the NIH in 2019, I finally had time to actually write a book on the science of intermittent fasting, which I did. And there's been a number of review articles. I did one with a former postdoc of mine it was in the New England Journal of Medicine and so on. But these are some of the effects, beneficial effects of intermittent fasting on different organ systems in laboratory rodents and humans. And I'm not going to go over these in detail, but in general, and I should point out, these studies are mostly, except with very rare exceptions, done in adults. They're not done in humans and children or the elderly people. They're not, some of them are done in normal healthy weight humans, and everything there looks good. both men and women of normal weight, certainly if they're obese, uh, overweight, there's definitely beneficial effects. The extent to which intermittent fasting benefits the health of individuals who are already in really good shape and have low body weight, it's unclear. We think there's still some benefits, particularly for the brain. Okay, and then the general working model that we developed for how both exercise, which there's a lot of work done on, and intermittent fasting benefit health over time is that each of these are challenges, they're stresses on our system, uh, probably on all our organ systems. And during the challenge period, during the fasting, during the exercise, cells activate adaptive stress response signaling pathways that upregulate things like antioxidant defenses, autophagy, DNA repair, uh, and others. I mentioned heat shock proteins, for example. Uh, but also during the fasting and exercise period, other pathways, particular pathways involved in cell growth and plasticity are downregulated. The major pathway that's been studied there is the mTOR pathway. Okay, and then during the recovery period, eating, resting, sleeping after exercise and fasting, during that recovery period, pathways are activated in cells that promote growth and plasticity, mitochondrial biogenesis, the increase in number of mitochondria. And then over time, over periods of weeks, months, years, this switching back and forth between the challenge and the recovery, and then repeating that over time, leads to long-term health benefits. All right, so that's the idea. I'm actually working on a third book now. I sent it to the publisher. It's out for review now. It's called Challenge, Recover, Repeat. And it just it's a long book. It's going to go into the science of this. Okay, I mentioned this before that this ketone beta hydroxybutyrate, which is elevated during the fasting period, acts as a hormone. This is from a recent review article I wrote. And it can activate certain transcription factors. There's also some evidence, although more needs to be done, that this ketone can modify proteins on lysine residues. So you've all heard of protein phosphorylation. You've probably heard of, I hope, protein acetylation, which occurs on lysine residues. So molecules can be added to or removed from certain proteins, certain amino acids in proteins, and the removal or addition of those low molecular weight molecules can affect the protein function. This may be true with this ketone as well. On the right, this just shows some of the data from my lab and another lab where both labs found kind of coincidentally, without knowing each was working on that this ketone upregulates BDNF expression in the hippocampus. Now, the brain, neurons in the brain can use either glucose or ketones as an energy source. Steve Canaan up in Canada showed using PET imaging with radiolabeled glucose or ketone that brain cells they're very plastic in their ability to use either glucose or ketone. So if a person is fasting, the brain cells are using ketones after the metabolic switch occurs and the blood ketone levels go up. It's the same with ketogenic diets, that's shown here, where after they're being on the ketogenic diet, the utilization of glucose goes down and the utilization of ketones goes up. And there's a perfect correlation between the blood ketone levels and the utilization of ketones by brain cells. And this is why you can, during Ramadan, you're only fasting for what, 12 to 18 hours, depending where you are on the planet or whatever. But you can fast for 40 days, which is like, I guess, another fasting thing in some religion. And you'll be alive. Uh, and the reason is because you're using, your brain cells are using, and other cells are using ketones from your fat, right? So your fat stores. And I guess one advantage of being overweight would be if we do have apocalypse and there's no more food available, you would live a little bit longer than everybody else before that our, you know, humans are completely wiped out, which I hope we don't do the way things seem to be going in the world. We showed that this ketone, beta hydroxybutyrate ester, can, if we supplement the diet in our 3xTG-AD mouse model, the ketone supplementation has beneficial effects. And then finally, this is my last slide, few slides on intermittent fasting, then I'll quickly talk about exercise and finish up. We finally did a study, and it actually didn't get published till after I retired, at the National Institute on Aging, looking at effects of intermittent fasting on the brains of humans who are at risk for cognitive impairment and Alzheimer's disease because of their age. They were between 55 and I think 72 years of age. They had obesity, body mass index at or above 30, and they had insulin resistance, done with a clinical way you do that with HOMA-IR or glucose tolerance test. Okay, so they were randomly assigned to 5-2 intermittent fasting, which I've talked about before, and what's called the USDA, U.S. Department of Agriculture, Healthy Living Diet. Or this may not be the USDA diet with our current administration in power. It may be eat only meat and, I don't know, and overeat protein, which actually too much protein is not good for you from the standpoint of aging anyway. Okay, but anyway, and then what we did is, before we started them on the intermittent fasting or the healthy living diet, we did, and when I say we, I mean, is someone asking me a question, or just they didn't mute?
No, they didn't mute. Yeah.
Okay. Right. So Dimitros Kapagiannis was the neurologist that was the principal investigator on this study, and him and the people working for him actually did all the hands-on work. My main role was in developing the protocol and being a cheerleader, and then going over the data and so on. Okay, so we did cognitive testing, functional magnetic resonance imaging of the brain, structural MRI imaging of the brain, did a lot of blood work, took cerebrospinal fluid to measure various things, amyloid beta peptide, phosphotau. There's other things that are being measured, have been measured that aren't in this published paper, like BDNF, for example. Okay, so what we found is that both interventions, the participants lost significant amounts of weight. The improvements in glucose regulation were greater in those on intermittent fasting compared to the healthy living diet. That's its HOMA-IR measurement and the hemoglobin A1C. This is a kind of the clinical evaluation for diabetes. Ketones were up. Interestingly, they were up a little, although I didn't reach significance in the healthy living diet, but they're significantly up with the intermittent fasting, and there are improvements in cholesterol and lipid profiles. Then the cognitive testing was very revealing. There were significant improvements in short-term memory, verbal fluency, executive functions, set shifting. So this is essentially task switching, being able to switch your attention from one task to another, which is something I'm finding for me personally is declining as I'm getting older. I think I've also noticed that in some cases, I kind of notice my cognitive function is going down. Actually, this is interesting. If you look at this, we already know this from cognitive testing that beginning in the late 30s, early 40s, performance on cognitive tests goes down. Some of these same tests shown here. So most of you, I guess, listening to this aren't over 40, but this is a good reason for you to think about what I'm presenting in terms of your rate of decline after you reach the age of 40 in cognitive function and what you might do in terms of lifestyle to sustain it, prevent the slowdown. And there were some interesting, small but significant amelioration of, well, I can't say that. Essentially, reductions in gray matter volumes in certain brain regions. Okay, exercise. This slide is from a review article by Henriette van Praag, who was a independent investigator in the Laboratory of Neurosciences at NIH where I was. Before I recruited her to NIH, she had discovered that exercise stimulates neurogenesis in the hippocampus of rats. And she did that work when she was at the Salk Institute. And then in my lab, again, she was independent of my own group, my own people. The organization of NIH is a laboratory at NIH is equivalent to a department at a university, and a lab chief, which I was at NIH, is equivalent to a chair person of a department at a university. So Henriette was a tenure track investigator in the laboratory or Department of Neuroscience. We collaborate on some studies, but mostly she was independent. So she showed at NIH, she did these elegant studies showing the time course of innervation of newly generated neurons in the hippocampus by inputs from other neurons within the hippocampus and outside of the hippocampus. So essentially, she showed how these newly generated neurons coming from stem cells are integrated into the circuitry in the brain and then provided evidence explaining how these newly generated neurons can improve or play a critical role in the improvement of spatial learning and memory conferred by exercise, regular exercise, at least in rats and mice. Now, one thing that's interesting from this, so exercise stimulates neurogenesis, synaptogenesis. It will upregulate neurotrophic factors. Interestingly, similar to exercise effects in muscle, exercise will increase angiogenesis, the growth of blood vessels, so will increase perfusion of the brain tissue. There have been many studies now identifying factors released from muscle cells or other cells in response to exercise that may play roles in enhancing brain plasticity in response to exercise. And some of them are shown here. Cathepsin B was one identified by Henriette von Praag's group, interleukin 6, irisin, the ketone BHB or BHA, beta hydroxybutyric acid. I mentioned it, so it comes from the liver. I mentioned that. It acts as a hormone. We know it upregulates BDNF, so it could be, we know BDNF is upregulated in response to exercise. There may be some factors coming from fat cells. Okay, human studies done by, for example, Kramer and others have provided evidence then in adult humans, including humans getting in the age of 60 years of age, aerobic exercise, but not stretching. We don't know about resistance training, but aerobic exercise can actually prevent decline in size of hippocampus. And that's shown from this study. This study showed that there's a correlation between VO2 max, which is an indicator of fitness, endurance fitness in particular, and BDNF levels in the cerebrospinal fluid and cognition. Okay, so those are the data from that, and there's the reference. I'm going to provide the slides, which will be distributed to all of you if you want to look at the slides. And I guess this is being recorded, so if you want to come back and look at it there, you can. So this is the general idea that intermittent fasting, exercise, and intellectual challenges all impose mild but beneficial stress on cells, and that mild stress upregulates pathways that promote stress resistance, but then as long as there's a recovery period, growth and plasticity. I'm going to skip, that's a repeat. I just want to show you one other interesting story before I quit, and that is has to do with enzyme in the mitochondria called sirtuin-3 or SIRT3. It's a protein deacetylase. So this enzyme removes acetyl groups from lysine residues in apparently at least 100 or more proteins in the mitochondria. Some are proteins in the electron transport chain. And we and others have found that another one is superoxide dismutase 2, SOD2, which is a major antioxidant enzyme in cells. It essentially converts superoxide generated during electron transport, oxidative phosphorylation. It converts superoxide to hydrogen peroxide, which is then further acted on and any potential toxicity removed. Okay, we also found in neurons that that cyclophilin D, which is a protein that's a component of the permeability transition pore involved in apoptosis, programmed cell death. We found that cyclophilin D is deacetylated by SIRT3, and that we think, or we didn't conclusively show that, we think then, based on other evidence, may protect cells against apoptosis. Certainly upregulation of SOD2 activity would. Okay, so in this study, we used CERT3 knockout mice. We found by immunoblots that there's hyperphosphorylation. These are blots using an anti-acetyllysine antibody. So an antibody that recognizes acetylated protein acetylation. So there's increased protein acetylation in the whole cells and the mitochondria in the SIRT3 knockout mice. And then this is we do immunoprecipitation with antibodies against SOD and so on to show that they're a target. And then we did both cell culture and in vivo studies in the cultured cerebral cortical neurons from SIRT3 knockout mice. They are more vulnerable to excitotoxicity, shown here, with glutamate, the endogenous excitatory amino acid that can also kill neurons, and then cadenic acid and NMDA excitotoxins in this study. And then we showed that if we overexpress SIRT3 in the neurons using an AAV, so if we increase SIRT3, they're resistant to excitotoxicity. We found in vivo that hippocampal neurons from SIRT3 knockout mice are more vulnerable to excitotoxicity in the seizure model. We found that in this study that physical exercise, shown on the right. So this is running wheel exercise for several weeks will increase SIRT3 protein levels in the cerebral cortex and hippocampus. And we also found, interestingly, in the cultured neurons that if we stimulate them with subtoxic levels of glutamate, so if we excite them a little bit, but not too much, SIRT3 levels go up. So this could possibly be relevant to normal increase in activity during things like learning new tasks and so on. And we also found that, so SIRT3 levels increase with exercise. And we found if we block glutamate receptors, specifically the NMDA receptor with this drug MK-801, then the ability of exercise to upregulate SIRT3 is abolished. So this shows that activity in the hippocampal network is mediating increased SIRT3 expression in response to exercise. When you exercise, like if you go on a run through the woods, which I'm looking at now where I live, not only will activity increase in neurons in your motor, sensory motor system, but also in your hippocampal prefrontal cortex. And I think actually this may be a reason that a lot of people find they do a lot of good thinking if they go on a long walk. I used to come up with a lot of good ideas going when I was on a long run. From an evolutionary perspective, that makes sense that as you're physically exerting yourself in your environment, it's important to remember where you're going, what you're seeing, whether it's a food source or a predator, where the predators and this and that. So I think like moving through your environment while you're exercising is probably really good for your brain. That's just hypothesis. Exercise increases the number of mitochondria in muscle cells over time. That's well established. We found that BDNF, which is induced by exercise, somewhat with intermittent fasting, and also with environmental enrichment, cognitive challenges, we found BDNF stimulates hippocampal neurogenesis. I'm not going to go into any more detail. This is a nice paper by Wu Cheng, who was a really good senior investigator in my group. she did many really nice studies. In this study, she showed in cultured neurons that as the neurons grow in culture and form synapses, the number of mitochondria in the neurons increases and ATP levels increase. This just shows that. So mitochondria are distributed throughout the dendrites and axons, and they're present right close to synapses where there's a lot of energetic stress. So if we knock down PGC1 alpha, which is a master regulator of mitochondrial biogenesis using RNA interference, then that reduces or blocks the increase in numbers of mitochondria and reduces synapse formation in these developing neurons in culture. And that's quantified here. And then if we knock down PGC1 alpha in hippocampal neurons in the adult mouse, and then let them go for a number of weeks and then count number of synapses, there's a significant reduction in synapses. So the conclusion of all this is that BDNF induces mitochondrial biogenesis. In culture, mitochondrial biogenesis is necessary for BDNF to promote synapse formation, and in vivo, mitochondrial biogenesis is necessary for the maintenance of synapses. And then I kind of got these out of order. We also showed that intermittent fasting upregulates SIRT3 expression, and interestingly, I mentioned that intermittent fasting is very protective against excitotoxicity. In this study, we found that intermittent fasting enhances GABAergic tone. So the main circuitry throughout the brain is excitatory glutamatergic neurons and inhibitory GABAergic neurons. And we're finding intermittent fasting enhances GABA tone and essentially preventing aberrant neural activity such as occurred during a seizure. Okay, I'm gonna quit. So this kind of a summary. The idea is challenge, recover, repeat. If that's done over long periods of time, that optimizes health. The three lifestyle interventions that there's really strong evidence are beneficial for the body and brain are physical exercise, intermittent fasting, and regular participation in intellectual and social endeavors. So keeping your mind, exercising your mind throughout your life. Yeah, I was told that it's okay if I run over a little, so I want to have time for questions. I can stay for another like 20 minutes after I'm done talking to answer questions. I wanted to point out that in the United States, and this is true now in some other countries, there's particularly capitalistic countries where there's a lot of forces, if you will, promoting poor health. Not necessarily the people working for those industries, aren't concerned about health, but so those are in monoculture agriculture, growing only corn, soybeans, and wheat, and then taking those grains and processing them, oftentimes getting rid of good things for our health and adding bad things like high fructose corn syrup, which is a big bad thing for health. Ultra-processed food industry where there's chemists who get paid to make foods addictive so that you want to keep eating more. And in fact, these are addictive. These foods are just like the same circuits and changes are occurring in their brain as you see with opioid addiction, drug addiction, gambling addiction, other things. The pharmaceutical industry is not all good. They do make some drugs that certainly help people. For example, vaccines, drugs for high blood pressure, some other things. But on the other hand, they make money
Beğenmedin herhalde sen.
by treating sick
Böyle
people.
işte onu açan bir şey yok.
Treating sick people. And so the point is that there's a lot of disincentives for having healthy lifestyles in the United States and elsewhere. And we, as a country, have been very bad in extending our bad health habits in terms of food and maybe drugs to other countries. So, for example, fast food, right? McDonald's, Kentucky Fried Chicken, Taco Bell, whatever, high fructose corn syrup, that really started here. And then it's the same with smoking and cigarettes, tobacco industry. United States was a big driver. We've actually improved it here, but now there's a problem in Asia. So there's a lot of, but I have some ideas for how this could change. Whether it will or not, I don't know. I'm not that optimistic given the for-profit motives of not only the companies, but the people in Congress. The legislators are often being lobbied by these, and they make money. So it's a problem. But the only good thing is that individuals can take responsibility for their own health. It's possible. They can eat healthy diets at relatively low cost. They can certainly exercise, and they can certainly keep their minds active. So here's an example from my group at NIH a while ago, and this is from a symposium that was organized by people from my lab when I retired. And this is what I've been doing since I retired about 6 years ago. I wrote 2 books. If you want to know about the brain, this is actually not a bad book because it's essentially looking at the brain from this perspective of glutamate, which is 90% of the neurons in the brain use glutamate as their neurotransmitter. And this is the podcast I do where I talk to some expert in neuroscience about their work. So thank you, and I'll take questions now.
Thank you very much, Professor Mattson, for an enlightening and inspiring presentation. Now, we would like to proceed with Q&A session. Dear participant, please feel free to type your question or comments in the chat box or use the raise hand option if you would like to speak directly. Yes, Ace, you can ask your question.
Thank you, Talha. Hi, Professor Mattson. Thank you for a lovely presentation. I have a background in psychology and I recently graduated from my master's in bioinformatics, and I'm also an athlete for 12 years now. And I really excited for the BDNF part for this presentation because BDNF was one of my special interests since it was extensively studied in athletes and clinical groups, especially. And I wanted to ask you about the valine 66 methionine polymorphism and its relationship with intermittent fasting and exercise in clinical groups or healthy people. Do you have any information about this part? Because I know that some variations of this gene affects the phenomena we observe, like psychological resilience or physiological resilience or something else. I don't know. Do you have any information about the variations of BDNF gene and intermittent fasting and exercise?
No, that's an excellent question. So this variation in BDNF sequence has been pretty clearly associated with risk for depression, right? Clinical depression. And as far as I know, I guess I should know this, right? But I'm not aware, certainly in humans, where this has been looked at. In other words, what you're asking is, uh, so exercise has an antidepressant effect, right? So one question is are people with a variant that increases their risk for depression, do they respond to exercise just in terms of antidepressant effect, just as well as people that don't have that variant? And then the same with intermittent fasting. Um, I'm not aware, but it's a really good question and thing to pursue. So that's all I can say.
Thank you so much. Also, I have another question for neurodevelopmental disorders like autism and ADHD. Do you have any insights for us, young neuroscientists or want to be neuroscientists, that intermittent fasting and exercise with the in relationship with neurodevelopmental disorders? In terms of research perspectives, how do we approach these topics? Like, I believe that body is a whole, and we forced to specialize in certain aspects of our fields. So it is hard sometimes to see some of the details that we might miss um, because we are so specialized. So you are an experienced professor in your field. So do you have any suggestions or any insights to give us?
I kind of do. You made me think of an interesting thing. So these are bits for a horse, right?
Yes.
Okay, I grew up on a farm, and my grandfather, my father, and therefore me and my brother trained and raced harness horses. Okay. And one thing I noticed very clearly is that, and I grew up in Minnesota where the winters are we get a lot of snow. And um, so we don't train the horses in the winter, right? They're mostly in the barn, in their stall. And so it's very obvious when they don't get exercise, their mood changes a lot, and they're more ornery and cranky. And then I noticed this too with our children, both our son and daughter. They ran cross country, and I was actually coach for the high school that my daughter ran at. And during the cross country season, their mood was much better. They're better able to focus on task in school and home, better able to do their homework without getting distracted. So from the standpoint of autism, which you kind of started your question with, and this is just kind of looking at this at a superficial level, but sometimes that's a good thing to do. So if and how much does exercise help children with autism or ADHD for that matter, right? And it seems like it probably should. And so we know that as far as risk factors for autism, one that seems clear is that if a woman has obesity or if they have some infection that causes systemic inflammation, but obesity essentially is systemic inflammation. Women with obesity, diabetes during pregnancy it's they have a there's an increased chance of their child being diagnosed with autism. And so it's important for women during pregnancy to be physically fit. And this, in some sense, goes against kind of the doctor, OBGYN doctor that the woman goes to. They may tell the woman, okay, you're pregnant, you need to eat more, maybe, and don't exercise too much because you have to have energy for the baby. But if you think about that from an evolutionary perspective, it doesn't make any sense. I showed you a picture in my first slide of hunter-gatherers, right? Would we be here, you and me, would we be here if, you know, they couldn't have healthy offspring under conditions of food scarcity or relative food scarcity, certainly not food overabundance. So I think that's kind of one thing. We know that's changed for heart disease, right? It used to be a cardiovascular doctor would tell their patient, okay, you had a heart attack. You don't want to stress your heart. But that's not true. It's actually good for someone who's had a heart attack to slowly do exercise because that improves their chances of having another heart attack. Yeah, so I don't know if that helps you any, or I've often benefited from looking back, kind of looking at evolutionary perspective or kind of move away from the nitty-gritty of a signaling transduction pathway or, whatever gene expression and so on, and look more. This old saying, think globally, act locally, but then come back to the global perspective.
Thank
Okay.
you so much.
Yeah. Any other questions?
We have a question in the chat box from Eda Aslan, who is a neuroscience PhD student with physiotherapy background. She's curious about aerobic exercise types and how and or if their effects vary in the parameters you mentioned, like neurogenesis, neural function, hippocampal volume, and etc. Is it just the aerobic exercise, or types like HIIT or MICT differ in results? Would you suggest a hot research area in this field?
More work needs to be done. There are definitely benefits of resistance training, obviously, for maintaining muscle mass, particularly as you get older. There is evidence that for production of exerkines in response to resistance training. So I think overall, aerobic exercise, I think, is better for cardiovascular health and regulation of blood glucose, so metabolic health in general. But the obvious thing would be to combine, do aerobic exercise, but also at least a couple days a week do some resistance training, which is one thing I did not do, and I wish I had. I used to run a lot, distance running, and back when I started running in the 1970s, the elite runners, the best runners in the world, all the runners, their training was running only. They didn't do any resistance training, working various muscle groups. And I had an accident where I tore some muscles from my inner thigh off the pubic bone, the adductor muscle, and probably, and I was doing something else besides running. When I did that, it could be that because I didn't do like this cross training type thing, that I was more vulnerable to that kind of injury. although I have really good aerobic fitness. The problem is this applies to both exercise and intermittent fasting. Clinical studies are not done where two different types of exercise, aerobic versus resistance training, or intermittent fasting, daily time restricted eating versus 5-2 intermittent fasting are done in the same study. You know, there's the study, usually the studies are you look at one intermittent fasting intervention versus no intermittent fasting, or you look at aerobic exercise versus no exercise, or like stretching, which isn't really exercising your muscles that much. But looking at multiple exercise or multiple intermittent fasting in the same studies would have to be done to make any firm conclusions.
I think we have another question from Fatma Nur. You can ask your question.
My question is a bit personal. As a psychology student who is aiming for a neuroscience master and after a PhD, what can you suggest me to graduation?
Sorry, say that again.
As a psychology student who is aiming for a neuroscience master and after a PhD, what can you suggest me to do before my graduation from university?
What should you do before your graduation? I'm not sure I understand the question.
What can I do for preparing myself for a neuroscience master?
You're undergraduate now?
Yes.
Well, at least in the United States, besides your grades, your academic grades and your coursework, it's important to show an interest in research by, volunteering to work in laboratories at your university, or, if you're clinically oriented, at shadowing doctors, that kind of thing. Yeah, those seem to be like the two key things, your coursework plus, and then your letters of recommendation are very important. It's important, ideally they'd be from, an accomplished scientist, not necessarily just a teacher of a course, but, someone who's actually, you've demonstrated by working in the lab or whatever your interest, and they should give you very positive. So there's the kind of the three things: coursework, letters of recommendation, and demonstration of your interest in pursuing a science career by doing volunteer work in labs or something else related to that.
Thank you so much.
Yeah. Okay, enjoy Ramadan. Don't overeat during the dark hours.
Yeah, noted. Yeah.
And
Thank
try
you.
to get some exercise. It's not all fun.
Thank you so much, Professor.
And then keep doing it the other 11 months out of the year.
All right.
Okay.
Thank you so much for participating in our seminar. If you'd like, we can take a photo, and some more participants can, if they're available, open their cameras also.
You're going to take a photo on the screen?
Yeah, screenshot.
Oh.
Yeah.
Okay.
Okay, I got some. Thank you so much, Professor. It
Okay.
was so lovely.
Have a good rest of the Ramadan and year.
Thank you.
Bye.